All right. Sorry I'm late. My lab meeting went a bit long. So, as a reminder, there's an exam review this evening from 6:30 to 8:30. It'll be in GN 101, so there'll be plenty of room. I will be going over the most important, complicated topics that you're going to see on exam 2. So obviously I will be covering some chapter 8A and 8B stuff tonight as well. The Thursday SI session, I'll cover it in more detail, but for tonight I'm just going to go through and give you the most important stuff that you're going to need for the exam. It will be recorded like always, but again, because it's a 2-hour session, it usually takes a while for it to upload to YouTube. So if you can attend tonight, I highly suggest that you do. Other stuff, I'll post a chapter 8 kind of study guide rather than a worksheet, since we're doing it a little bit out of order because of this. So I'll post that before Thursday's SI session. And let's see if there's anything else. I guess that's it for now. So we'll continue on from last time, speeding right along. Last time we talked about how to label and identify disaccharides. Specifically, the example we started with was lactose. And last time, we drew out lactose, and it looked like this. On the exam, ignore this thing right here, and this is a squiggly line. On the exam, if given this structure, you won't be asked to label it as lactose. More likely, you'll be asked to give the long form name of this, which involves identifying the component monosaccharides, identifying what type of linkage connects them, and what numbered carbons make up this linkage. So last time we found out that this monosaccharide is what? Let's see, anyone remember? It is galactose, and the quick way to identify galactose is to compare it to glucose. So glucose should be the easiest structure you can identify regarding the monosaccharides, because glucose will have this pattern for functional groups. You see how I've drawn the functional groups alternating up, down, and the squiggly one doesn't matter? That is glucose. Galactose is easily identified by comparing this thing to glucose and noticing that only one spot is different, which is this spot over here on the left. If you look at this spot over here on the left, the hydroxyl in glucose is pointing down, but over here in galactose is pointing up. That's the only difference between them. So it's a quick way to identify galactose, because glucose is easy to memorize, and galactose is just one small change. This one over here, what did we identify it as? This one over here is actually glucose, exactly. And how do we know it's glucose? Well, let's look at the functional groups. Pointing up, pointing down, pointing up, pointing down, and that one doesn't matter. It is that alternating pattern here, so this one is glucose. The next important thing we identified last time is what type of linkage connects these two monosaccharides together. So you see these elbow turns, these bends here? They do not represent CH2s, like this part here. These bends here do not represent CH2s like you normally see in organic chemistry. They're just a notation, an old-fashioned notation to indicate a beta linkage. And it is a beta linkage because if you look at the arrangement here, over here on the left, this functional group is pointing up, and the CH2OH is pointing up. Review previous notes or the previous information, you'll notice that when this functional group and the CH2OH are both pointing up, that is a beta linkage. If they are pointing in opposite directions, that would be an alpha linkage. So far we have galactose, beta, glucose. The last thing we have to identify are what numbered carbons make up this linkage, meaning what numbered carbon is this thing on the left and what numbered carbon is this thing on the right, which we went over last time as well. The way you number monosaccharides is you want your most oxidized carbon to be as low of a number as possible. In organic chemistry, you learned, oh, your alcohol function group has to be as low of a numbered carbon as possible, or your carboxylic acid has to be the lowest numbered carbon possible, right? Those are your organic chemistry rules. The actual reason why that is, the more general rule, is your most oxidized carbon needs to be as low of a number as possible. So on galactose here, our most oxidized carbon is this one. How do I know? It's the only one bonded to two oxygens. Oxygen 1 right there and oxygen 2 right there. So because this is the most oxidized carbon, I need to label that with as low of a number as possible. I'm going to label it with carbon 1 in this case. Most of the time, carbon 1 will be the answer. Let me just check that I labeled it correctly. If I call that one carbon 1, this one must be carbon 2, this one must be carbon 3, carbon 4, carbon 5, and carbon 6 is outside the ring. So galactose, the beta linkage starts at carbon number 1. Okay? Let's see what numbered carbon it is on glucose. So same rules apply. You want to number your carbons starting with the most oxidized carbon being as low of a number as possible. The most oxidized carbon is this one because it's the only one bonded to two oxygens. And so this one ends up being carbon 1 over here. That means this is 2, this is 3, this is 4, this is 5, and the sixth one is outside the ring. You'll see that happen a lot. So therefore, carbon number 4 on glucose is a component of this polysaccharide, is carbon number 4 here. Okay? So our full name and the answer to this question, if it's presented to you, they'll give you this structure. What is the name of the structure? It is galactose beta 1,4-glucose. Of course, the short name is lactose, but the answer choices will have that instead. So, some unique things about this disaccharide. Lactose is the only beta-linked sugar humans are able to digest. And even then, not all humans are able to digest lactose, right? You've heard of the phrase lactose intolerance. Those are people who do not make the enzyme lactase and are unable to break down lactose because they can't break this beta linkage. It turns out in nature, at least regarding mammals, we are really bad at breaking down beta-linked sugars. The only one that we sort of have the capability of breaking down that is a beta-linked sugar is lactose. So that's a very unique feature of lactose. Only beta-linked sugar we can digest. And even then, not everyone can. Lactose intolerant people are unable to. All other sugars that we process, all other polysaccharides and disaccharides that we are able to digest will be alpha-linked. If you see any other sugar other than lactose that is beta-linked, any other polysaccharide that is beta-linked, note that we will be unable to digest it. So I'll go over one more disaccharide example. And in fact, let me use the picture here so we can go fast. All right, don't look at the answer. Ignore the caption, close your eyes. Okay. All right, so you're given this as your disaccharide. Same rules apply. You don't need to give me the short name here. You got to give me the long disaccharide name. So the first thing we're going to do is we're going to identify the monosaccharides. Can someone tell me, or as a group, what is the monosaccharide on the left? It is glucose, and we can easily identify glucose because of this alternating pattern, right? Up, down, and the anomeric carbon doesn't matter. So that is glucose. What about the one on the right? What is that one? It is also glucose. Note that it is flipped around from what you guys are probably usually used to, right? Where's the CH2OH this time? It's down here. Okay, that's fine. That still makes it glucose. This is CH2OH pointing up. That functional group is pointing down. That one's pointing up. That one's pointing down. So it's an alternating pattern again. So we're naming this thing. The first thing we're going to look for in our answer choices is glucose, something, something glucose. Okay? Next, we have to investigate what type of linkage connects these two things together. And you know, starting on the left is always the easier way of doing it. Let's see, this linkage, and notice there's no bends this time. This linkage here is pointing down compared to the CH2OH that is pointing up there. What type of linkage must that be? Alpha linked, because they are pointing in opposite directions to each other. So, multiple choice answers, we could probably narrow it down to whatever says glucose alpha blank glucose. The last blank is what numbered carbons actually connect these two things together. So, we'll start on the left. What numbered carbon is this one right here on the left here? Carbon 1, because it is the most oxidized carbon in this glucose over here. What about the one on the right? It is also 1. Be very careful that glucose is flipped around this time. It is still carbon 1. How do I know? Well, this carbon over here on the right is attached to 2 oxygens. So it is carbon 1 for the glucose on the right here. That is why the full name of this, for those that were closing their eyes when the answer was put in front of you, is glucose alpha 1,1 glucose. That would be the answer given here. This is a special disaccharide with the given name trehalose, and it has some interesting properties. I'll have to look at the exam again. This might be one of the few disaccharides you have to know the specific details of. I'll give it to you at the exam review if it turns out that it is. But bottom line, you're given some disaccharide you've never seen before, you can still identify it in the process that we've done here. Some other questions could be asked about this, such as, there's many different ways it could be asked, is trehalose a reducing sugar? Is trehalose a reducing sugar? Well, raise your hand if you think yes. Raise your hand if you think it is not a reducing sugar. And raise your hand if you have no idea what I'm talking about. All right. So to remind you, the phrase reducing sugar is one of like 4 different ways to give you a complicated way of asking you, does this sugar have a free anomeric carbon? Which we went over we went over the steps on how to identify a free anomeric carbon, which we're going to apply here. If this sugar turns out to have at least 1 free anomeric carbon, it means it is a reducing sugar. If it doesn't have a free anomeric carbon anywhere at all, we are not going to call it a reducing sugar. Okay? So, what a free anomeric carbon is first you have to look at the anomeric carbons. The anomeric carbons are the ones bonded to 2 oxygens, which we've identified already, right? That's this carbon on the left, I'm circling right here. That is one of the ones bonded to 2 oxygens. And this is the only other one bonded to 2 oxygens. So we've identified the anomeric carbons, or these 2 right here in the center. The question now is, are they free anomeric carbons? A free anomeric carbon is when these carbons here have a hydroxyl group attached to them, have an OH group attached to them. Do we see any OH groups attached to either of these 2 things? No. What has happened here is that the OH groups at each of these anomeric carbons were fused together to form this glycosidic bond between them. There are no free anomeric carbons in this picture. Therefore, we can make several conclusions. One is the question that I gave you, is that this is not a reducing sugar because there are no free anomeric carbons. Another conclusion you can make is that this sugar cannot undergo mutarotation. What mutarotation is that a free anomeric carbon, the functional group can rotate from pointing up to pointing down, meaning it can freely rotate from alpha to beta and back and forth conformation. That only happens at free anomeric carbons. Because there are no free anomeric carbons here, it cannot undergo mutarotation. It is not a reducing sugar. All right, so that's many different little questions they can ask regarding this or any disaccharide given to you. All right, so since we're a little bit short on time, I'm going to move right along. But that is the basics of disaccharides. Okay. Oh, that is the end. Awesome. All right, I'm going to move on to 7b. Okay, so 7b covers polysaccharides. So 7a covered monosaccharides and disaccharides. 7b covers polysaccharides, of which there are several different categories. And really all you need to be good at is identifying just enough of a picture of a polysaccharide to know which one it is. You don't need to know every single position of every single oxygen, every single carbon, every single hydrogen, and double bonds and single bonds, and every functional group. That doesn't matter. You just need to identify certain characteristics that given any of these polysaccharides, you know what type it is. So that's what we're going to focus on mostly. So again, what is a polysaccharide? Well, we talked about monosaccharides and disaccharides. All a polysaccharide is a monosaccharide repeated a bunch of times. Most sugars are actually stored or have the structural component as a polysaccharide, and only under certain scenarios will you break down a polysaccharide into its monosaccharide components. So the polysaccharide form of sugars is the most preferred form. And you see some given examples here. Starch and glycogen are your two characteristic examples of energy storage polysaccharides. We'll see why in a bit. Chitin and cellulose are structural instead. And then there's a bunch of extra ones we'll go over at the end that serve a bunch of different purposes, but they kind of group them together as cell recognition or cell surface signaling molecules. So, and the phrases homopolysaccharide and heteropolysaccharide. Very similar to when you learned about proteins, homomer and heteromer with proteins, where back then, if it was a protein made of the same subunit, that is a homomultimer. If it was made of different subunits, it was a heteromultimer. Same logic applies here. A homopolysaccharide is made of the same sugar repeated over and over. A heteropolysaccharide is made of a bunch of different monosaccharides put together. So we'll start with starch. Let me get a nice picture of starch here. Starch comes in two different forms, in a mixture of these two different forms in solution. Starch can come in an amylose form and an amylopectin form, which you don't have to overthink it. The only difference between these two is that amylose is the linear form and amylopectin has branches in it. That's about it. What you do need to be able to identify are the kind of general features that make what you are looking at labeled as starch and not other polysaccharides. So the general features you should look at is, one, what is the monosaccharide making up this polysaccharide? Well, let's look at any one of these pink things. Let's look at this pink thing here in the bottom left. And we see the CH2OH pointing up here. They got really lazy. They stopped drawing in the rest of the functional groups. Down, up, down, and that is your anomeric carbon. What monosaccharide must compose starch? It is glucose, repeated over and over and over again. Now, that's the first thing you have to do, is identify what monosaccharide composes this polysaccharide. Next is what type of linkages bind them together. So let's look at amylose first, because it's slightly more simple. And let's look at the first two glucoses on the left here. We look at the linkage here, and actually I'll ask, anyone tell me, is that an alpha or a beta linkage? It is an alpha linkage, and we can quickly see why, because at this linkage, we see that this functional group is pointing down. Meanwhile, the CH2OH on the left there is pointing up. Because they are pointing in opposite directions, it is an alpha link. And it's the same alpha link you see the rest of this structure in this amylose form of starch. So starch, by definition, or at least the amylose form, is a bunch of glucoses hooked together via an alpha link. And in fact, it is hooked together via carbon 1 and carbon 4. And you can use the same procedure we used earlier to figure out what numbered carbons these are. Or you can choose to memorize it because it is blatantly stated in the notes for starch. Feel free to do whichever one you want. So, if you're given a polysaccharide, you know it's starch, and specifically that it is amylose version of starch if it is a bunch of glucoses hooked together via alpha 1,4 linkages. Let's contrast that with amylopectin. It is still starch, and in fact, if I zoom out a bit, this bottom part here basically looks exactly the same as amylose, right? This bottom line here is a bunch of glucoses connected to each other via alpha 1,4 linkages. The only difference with this form of starch is that every now and then there is a branch sticking out of that linear chain of glucoses. At what numbered carbon is this? That is carbon 6, right? The carbon that's sticking out of the ring is carbon number 6. So every now and then a branch comes out of the linear chain of glucoses, and you can start another linear chain of glucoses from this branch. So amylopectin, think of as a branching network of glucose chains, okay? There's a reason why you have these two different forms of starch, which I'll explain in a second. Yes, question?
You said that amylose was just a bunch of glucose held together by what?
Alpha 1,4 linkages. Yep, alpha 1,4 linkages. And amylopectin is basically the same thing, except it also includes alpha 1,6 branches. That's it. That's what this one spot here is an alpha 1,6 branch. And imagine that branch shows up later down the line, later on the illustration. You know, obviously they didn't have room to show it, but these branches show up every now and then. And there's a reason for that. When you have branched polysaccharides, it allows you to more efficiently store glucose in a compact space and more efficiently extract glucose out of starch, out of a polysaccharide. Because, and if I sort of illustrate this here. All right, so let's say we had amylose, which is a nice linear form. I'm zoomed out of that picture here. That all I can really see is a nice line of glucoses linked together. Like imagine there's a bunch of glucoses in this line. If I were to try to extract energy out of amylose, I could, if I were to try to extract glucose out of amylose, I could really only take off 2 glucoses at a time, right? I can't remove glucoses in the middle of this thing. I can only remove a glucose on the very end of it, which there are only 2 ends on the linear form of amylose. But what about for a branched form of starch? I'm just going to draw a bunch of branches here. When you have a bunch of branches in starch and any other polysaccharide, energy storage polysaccharide, you can remove a lot more glucoses at a single time point because each branch ends as well, and you can remove glucoses from each one. So the more branches you have in an energy storage polysaccharide, the more efficient you can store glucose onto it, and the more efficient you can extract glucose out of it because there's more places to extract it out of. So that's the key feature of branching in any energy storage polysaccharide. Okay? Again, bottom line, more branches, it is easier to extract glucose out of the energy storage polysaccharide. The reason why I say it as a general term for energy storage polysaccharide, because that property not only applies to starch, indicated in amylopectin, but it also applies to glycogen, wherever it is here. There we go, glycogen. Glycogen, and they don't even give you a drawing of glycogen because, plot twist, it looks exactly the same as starch, except for one difference. The only difference between starch and glycogen is that glycogen has more branches. That's about it. They phrase it slightly differently. They phrase it saying branches in glycogen are every 8 to 12 residues, meaning every 8 to 12 glucoses, there's a branch. In starch, I think it's a different number, 12 to 20. So you have to go a longer distance before you see a branch in starch. What that basically means is that branches are more abundant in glycogen. So, a question, which one, starch or glycogen, do you think you can more efficiently extract glucose out of? Glycogen. And guess what? Glycogen is the animal energy storage polysaccharide. Meanwhile, starch is the plant energy storage polysaccharide. So that makes sense. Animals need more energy and they need to extract it at a faster rate, so they're going to prefer to use glycogen. Of course, we eat, fruits and vegetables, so we do digest starch as well, but we store our excess glucose in the form of glycogen because it is more efficient for us. So that is glycogen. Don't worry about dextrans. That used to be asked about. I haven't seen it asked about in many semesters. When I look at the exam, if it happens to be asked about this semester, I will cover it in detail at the exam review. So we're going to skip dextrans. Okay, so after energy storage polysaccharides, we go into structural polysaccharides. So structural polysaccharides, there's really one significant difference. No matter what the structural polysaccharide is, there's one very obvious difference. And that is the type of linkage connecting a structural polysaccharide together. The energy storage polysaccharides, both glycogen and starch, were connected via alpha links, right? Which I said way earlier when we were talking about disaccharides, that we are able to digest alpha-linked sugars. So if we're talking about a structural polysaccharide, one that we shouldn't be digesting, it is no surprise that those are made of beta-linked sugars because we shouldn't be able to digest them. The example they give here in the bottom half right here is cellulose. Cellulose, it turns out, looks exactly the same as amylose. It is made of glucose, and the glucoses are connected at carbon 1 and carbon 4. There's just one small difference, is that cellulose has those glucoses connected via a beta link. Meanwhile, amylose had those glucoses connected via an alpha link. And that small difference gives you that difference between an energy storage polysaccharide and a structural polysaccharide. It's just what type of linkage connects them to each other. And they give you a couple examples other than cellulose. Excuse me. Other than cellulose. I gotta learn a sign language apparently. Other than cellulose is chitin, which they used to give a picture of it. Let me look real carefully. Yeah, there we go. Don't bother memorizing this complicated looking structure of chitin because all you really got to do is identify just subtle, small little details that if you see that detail, you know it's chitin immediately. Well, let's compare chitin to cellulose. Cellulose is easy. Cellulose is glucoses connected to each other via a beta link. What is the difference between cellulose and chitin? This random set of amide functional groups, right? You see this amino and carboxyl group attached to carbon number 2, I believe. It doesn't really matter what numbered carbon it's attached to. So if you want to identify chitin easily, just look for something that looks like cellulose, but there's a bunch of amide groups attached to it instead, and you're done. Then you know that's chitin. It is also a structural polysaccharide. We know that because it is beta linked to each other. And you can look in the notes and you find that chitin is a component of fungal cell walls and the exoskeleton of certain insects. So we see why it has a structural purpose, is it's built based off these beta linkages. There's a couple other polysaccharides that they have here that you can kind of diagnose what it would look like based off its name. These are very unlikely to show up on the exam, but if you see a polysaccharide you've never seen before, because it's a multiple choice exam, you can kind of guess at what it is. For example, this one up here, it won't be color coordinated on the exam. Using your knowledge of identifying monosaccharides from each other, if there is going to be chair confirmation on the exam, I'll explain it during the exam review. You would be able to identify that this thing is made of a bunch of mannoses. So instead of being made of a bunch of glucoses, this thing is made of a bunch of mannoses. And you can look for whatever multiple choice answer choice has the word that sounds like mannose in it, and you'll be able to pick that one pretty easily. There used to be another good example in here, which I can't see. This one isn't what I'm thinking of. I might have to give it during the exam review. Nope. Do they have a picture? Yeah. Sometimes there's a picture of agarose. And it turns out that agarose is actually just a galactose polymer rather than a glucose polymer. So all those features we talked about earlier regarding glucose polymers apply to agarose and agaropectin, which sound very similar to amylose and amylopectin, right? Amylose and amylopectin, we know what those look like, and we know they're composed of glucoses. If you see the phrase agarose and agaropectin, it's the same stuff, but instead of glucose, it's galactose instead. Okay? So there's little shortcuts like that. All right, so those are the two probably easier categories of polysaccharides to go over, is the energy storage ones and the structural ones, because they're very similar, they just differ in the linkages. Now we get to the less, simple stuff. We'll start here with glycosaminoglycans, which I'm going to simplify for you as well. So these are a special type of polysaccharide that you can easily identify by looking for two features. You will not have to name any of these. You will not have to know that this is specifically chondroitin 4 sulfate. You will not have to know that this thing on the right is specifically heparin. But what you do need to know is that these are collectively glycosaminoglycans, which you can identify via the following properties. A glycosaminoglycan is a heteropolysaccharide composed of a repeating pattern of 2 monosaccharides. So 2 unique monosaccharides are just repeated over and over and over again. So that's why you see in this picture, in each of these boxes, they only draw 2 monosaccharides, because it's those 2 that are repeated over and over and over again in each of these glycosaminoglycans. So that's the first thing to look for. The next thing are these weird looking functional groups here. You see an amino group here, and you see negatively charged functional groups on there. If you have both of those properties there, where you see some type of amino group somewhere on there, who cares where? And if you see negatively charged functional groups somewhere on there, who cares where? And that it is a repeating disaccharide, those are the 3 properties you need to identify this as a glycosaminoglycan. No other polysaccharide in your notes will have those combined properties. So again, you're looking for a repeating disaccharide, you're looking for amino groups on there, and the third one there, negatively charged functional groups on there. If you can find all 3, whatever structure you've never seen before, you can classify it as a glycosaminoglycan. And it is another category of structural polysaccharide. It's just a special one, not as simply identified as the other ones we've gone over. All right, next category of polysaccharides is glycoproteins. So a glycoprotein is a special type of polysaccharide that sticks itself onto a protein. It makes sense, it's in the name, glycoprotein, sugar protein. The reason why we do this is that when a protein is made in the body, when a cell makes a protein, it needs to know when to keep it around, where to send it around in the cell, and whether to degrade it, whether it's time to degrade the protein. They do so by attaching certain polysaccharides to them. There's 2 categories of glycoproteins, O-linked and N-linked glycoproteins, and they differ simply on what amino acids are associated with each one of these types. But again, if you want general functions of glycoproteins, all are right here. They all have something to do with protein stability by altering these properties of that protein. Your O-linked glycoproteins come in 3 flavors. Serine, threonine, and 2 flavors, serine and threonine, just connected in different ways. So an O-linked glycoprotein is easily identified by some type of sugars, 1 or more, attached to an amino acid. It is your job to figure out what amino acid is attached to it. But luckily you guys just took exam 1 and you totally remember all 20 amino acids. So the most important ones for exam 2, to help you along with that, are for O-linked glycoproteins. You're looking for ones attached to serines and threonines. And for comparison's sake, let me look. Yeah. For your N-linked glycoprotein, you're looking at one attached to asparagine. And the only example they have here is one attached to asparagine. So there's only 3 amino acids you have to worry about. If you see any one of those attached to a sugar, whether 1 or more sugars, you know you're dealing with a glycoprotein. Okay? So, now, talking about the function that I mentioned earlier, what glycoproteins do, the specific one that you may be asked about is its function to prevent the degradation of a protein or to signal when it is time to degrade a protein. And it is simply based off this sugar right here called sialic acid. Okay, so it's very important, this next statement here, and I'll break down this figure in more detail later. Sialic acid is a special marker on a glycoprotein that when it is present, it's telling the cell to not degrade the protein. Okay? Sialic acid is like a protective marker. If a protein has both of these sialic acid markers on there, the cellular mechanisms, the cellular machinery will know not to degrade that protein. But every now and then, due to whatever reason, one or both of these sialic acids can be removed out of the glycoprotein. And what ends up happening is that the cell thinks that it is time to break down that protein, and it will go through a whole, I think, lysosome-based degradation. I don't remember where. Somewhere in the notes they give you the specific details, but bottom line is if it's missing those sialic acids, the protein that the glycoprotein is attached to will be degraded. And you just need to be missing one of these. You don't need to be missing both of them. If at least one of them is missing, it will be degraded. So that is the specific protective effect of N-linked glycoprotein. Other questions they may ask regarding glycoproteins are just which type of glycoprotein are you looking at when given a picture. These are the only 3 types of glycoproteins you could possibly have or be asked about in this class. Let's look at the features associated with them. The easiest one to identify is the high mannose glycoprotein. Why? Look at all the mannose. Done. Next one, complex glycoprotein. This next comparison between complex and hybrid is a little bit weird, and sometimes it's hard for people to see, so I'll try to break it down. A complex glycoprotein is the one that has these 2 unique, diverse set of diverse composition of sugars in this branch form here. You see galactose, you see galactoamino acid or something, galactoamino sugar or whatever, mannose and sialic acid there. So it's a very balanced looking picture. That is what complex is. Hybrid is a little bit easier to identify because when you look at its 2 branches, one branch looks like high mannose and the other branch looks like the complex one. So how to identify each of these types of N-linked glycoproteins? 1, high mannose, very easy. It has a bunch of mannose in it. Hybrid, in my opinion, is the second easiest to identify because one branch has a bunch of mannoses in it and the other one doesn't. Therefore, whatever's left over, if you don't have either the high mannose or the hybrid looking picture, you have the complex looking picture. Yes?
Can those be O-linked?
These ones are N-linked. O-linked, I don't think he gives any examples like this. He gives you like a functional example of an O-linked glycoprotein, but you won't have to identify it in as much detail as the N-linked stuff was. O-linked glycoprotein, it gives you an example of a cell surface receptor protein. That's kind of what this picture is trying to show you, and I'll explain what this picture is showing you. Is that a cell surface receptor protein contains a bunch of different domains, a bunch of different areas to it. There is down here, this squiggly part down here is inside the cell. That is your intracellular domain of the protein. This stuff inside the cell membrane is your intermembrane space domain of the cell membrane. Then you have this long stem-like structure, and then you have this gobbledygook up here, which is your actual functional part of your cell surface protein. Is this globular head. Whatever signaling function the cell surface protein has depends simply on this. But notice that the rest of this protein's structural job is to get this sticking out above the cell membrane to do whatever signaling job it's supposed to do. In order to get it this high up and away from the cell membrane, you have this stem-like structure, which is coated in a bunch of O-linked glycoproteins to keep it stable. So that is what and the one example of O-linked glycoproteins he gives you is that you see it in a lot of cell surface proteins to construct this stem-like structure. And that's all we'll do for now. I will continue at the exam review, and I'll go over the specific stuff that you should be studying for the exam.